Hello, everyone, and welcome to phyloseminar.org. This is the second talk in a series on 3 talks on next-generation sequence evolution models. Please use the YouTube live comment box to ask questions. Today's speaker is Pierre Barrat-Charlaix. Pierre did his PhD at Sorbonne University on the topic of generative protein models. He then worked on the evolution of viral pathogens at the University of Basel in the team of Richard Nair. He's currently a research associate at the Politecnico de Torino in the team of statistical physics and interdisciplinary applications. He's interested in understanding evolution at the scale of proteins or of simple organisms. He tries to develop quantitative and interpretive models to describe this evolution. Welcome, Pierre, and thank you for participating.
Thank you. So thank you very much, Eric, for having me here. So the work I will talk about tonight is something about ancestral protein reconstruction using generative models, and more specifically autoregressive models. So the main idea here is that recently there's been a lot of developments on the statistical modeling of protein sequences. So the idea is that you can group proteins that share, so homologous proteins that share a common ancestor into families. And essentially, so with some caveats, but essentially proteins in the same family have a very similar structure and a very similar biological function, which can be a lot of different things. And the idea here is of this generative modeling is that this means that during evolution, there are strong evolutionary constraints on these proteins because they have to maintain the function, meaning that the members of the families that you may find in different species, if you align them, you will find some strong statistical patterns, like some amino acids that are completely conserved, some columns of the alignment that are very correlated, and this should be indicative somehow of function. And the idea of generative modeling in general is that you take these alignments, assuming that somehow you have information about function, and you try to learn what I call functionality landscape. So some function that tells you whether an arbitrary sequence of the length of the alignment would be a potential member of the family. So here in my representation, I see this as some kind of energy. So down is good. So you observe the blue points and you try to infer. So this is a high dimensional space, obviously. You try to infer this curve from the data. So there are different ways to do this, and I mean the point is not to explain all of those, but just to mention them. So the first things that really worked very well in this field is the POTS model, so where you essentially try to model both the interaction between different columns of the alignment, so different sites in the protein, and the conservation. So you do this with couplings and fields. What I will use here is a bit of a different architecture in a way. It's called autoregressive models, where the idea is that the first position, so let's say the first position of the sequence is independent from all others. So it has an independent distribution. Then the second one depends only on the first one. The third one depends on the first two, etc., etc. And so this way you also model some interactions, but only in a forward way. And then recently there has been some development with deep learning. So people use variational autoencoders and more importantly transformers. So things like MSA transformer, ESM. So you have a lot of ways to kind of infer these functionality landscapes. So we start to know quite well how to do generative models of protein sequences. So they're being used for a lot of things, and what I want to insist on here is that a key ingredient of all of these generative models is that they take epistasis into account. So they take into account the fact that a mutation at one position in the protein is, so the effect of a mutation depends on the context, depends on what happens in the rest of the sequence. So columns of the MSA cannot be considered independent, at least if you want to generate novel proteins. So this seems to be important for function. So the point here is, can we use these new developments for phylogenetic problems? And the problem here is ancestral sequence reconstruction. So the idea is you have this alignments. And so these are homologous proteins. So you have a tree behind this that describes the evolutionary history of the sequences. And the question is, can we infer the sequences at the internal nodes of this tree, right? So at the root here is maybe not a very well-defined node, but at least at the internal nodes here, given that we know the leaves. And so there are different applications. Some are, pure curiosity, say, so what were ancient proteins like? What was ancient life like? Then you have some maybe more practical question in a way. So you can study the sequence to function relationship, because if you have the sequences at the internal nodes, you can look at the mutations that happened on the branches. And maybe if the function changed slightly along the tree, you can, link these to specific mutations. So this is interesting. There is another application, which is a bit strange, which is that when people use typical algorithm to reconstruct these ancestral sequences, they tend to find proteins that are more thermostable than the present-day proteins. So especially if you go very far back in the tree. So this is one, in a way, application, like unexpected application. And so the question here is, can I somehow use what we've learned about these functionality landscapes, these complex models of protein, to infer these ancestors with more accuracy, okay? So this is essentially the goal. So before I explain how I do this, I just want to do a recap on what traditional algorithms are for ancestral sequence reconstruction. So here I will always be in a setting where I assume that the tree itself is perfectly known. Okay, so my problem is not to infer the tree. I assume that someone else did it for me. So you have a lot of software that does this kind of thing. And once you have the tree, so this is one of the two ingredients, the second ingredient that you need is a sequence evolution model. So it's essentially a model that tells you the probability starting from an ancestral sequence here and given some amount of evolutionary time, what's the probability of observing a given child sequence? So you can, in mathematical terms, it's a propagator, right? It's a probability of observing the child given the starting point, the ancestor, and the time delta t. And once you have these two things, you can, in principle, you can infer sequences at any internal nodes. So either the most likely sequence, or you can sample from some posterior probability, but you can compute everything. So how do you compute this? So the algorithm that you use is, so I never know what the official name is. I call it Felsenstein's algorithm. Then I think Felsenstein's maybe something a bit more specific, but I mean, I will explain what I do, and I think you will recognize that it's the same ideas. So the idea is that you have this tree and you have the leaves, S1, S2, etc., that you know. And what you want to compute, so this is the data, what you want to compute is the likelihood of having a given sequence X at this internal node N. And this likelihood is the probability of observing the data if this internal node was in state X, okay? And averaging over every other internal node. So the way, I think the easiest way to set up this problem is to decompose this into two likelihoods. The first is the one of the data that is below node N. So this I call the down likelihood, which would be the blue thing here. So it's the probability of the data below N if N is in state X. And so this has two, you do this in two steps. So if N is a leaf, then this is simply a delta, right? Because then X has to be in the state that you observe at this leaf. And if not, you can write some recursive relation. So essentially, it's the product of all the children of N of the probability of going from sequence X to the sequence of the child times the likelihood of the child, and you have to sum over all possible sequences at the child node, okay? So the sum here is over all possible sequences of this child node, and then you have, this only depends on what happens below the child node after this. And so this is done, computed. So assuming you can compute this kind of sums, then in a linear time, so in time linear in the number of nodes, you can compute this down likelihood, and you can know this you can take into account these blue leaves. And then you can do the same thing. So notation is a bit more heavy, but you can do essentially the same thing for the other part of the tree. So the up likelihood, which would be the likelihood given that the ancestor of n is in some state, the likelihood of the leaves that are above n. So this is computed exactly in the same way. You have a set of recursive relations. And what's important is you can essentially just combine these two likelihoods by doing one last sum, which depends on the branch length above n. So this combines essentially the blue and the information from the blue and the red leaves. And so this up likelihood you can also compute in time that is linear in number of nodes. And so you have finally a likelihood that is linear in number of nodes and depends only on the data, the tree, and the evolutionary model that you use. And obviously the problem here is that you have to be able to compute these sums. So you have to be able to compute to sum over all possible sequences at a given internal node. And if you take the general problem, this is very hard because you have a very high number of possible sequences. So you cannot do this numerically in general. So what people normally do is they assume that the sequence evolution model is position by position, is independent for every position. So all positions evolve independently. So they focus on one position, which here I take it to be nucleotides just because it's a bit easier to represent. And here, essentially, the sequence evolution model would be something that tells me if I know that I have some probability to be in a given state at some time, so which would be this P0 here, after time delta t, how did this distribution change, right? This is the sequence evolution model. And the traditional way to do this is to write a continuous time Markov chain. So what you do is you say from any given state, say A, I have transition rates, so rates of transition from this state to the other three states. So here these are three numbers. And since I have 4 possible starting states, I end up with a matrix that is 4 by 4 with essentially the off-diagonal terms, which are the interesting ones. And this matrix Q, so here it will be 4 by 4 if I have nucleotides. Of course, if I have amino acids, it would be 20 by 20. And here, essentially what you can say is that the derivative of this probability is equal to the probability times Q. And so this P delta T, so this distribution of states over states after some time, is the initial state times the exponential of the matrix Q multiplied by the time. And here mu is irrelevant, it's just a normalization factor. So this is the traditional way to do, and actually I will do almost like this. An important thing to mention is that, so I mean, this is theory of Markov chains in a way, and this is, it's very well known. I did not invent anything here. If you describe a model that is time reversible, then you can always write your matrix Q as the product of a matrix that is symmetric, which I call H, and a matrix which is diagonal, which has Q terms, where Q is so small q, which is the number of states, with values that sum to 1, positive values that sum to 1. And these two matrices have a very nice interpretation. So if you wanted to, build these evolution matrices from scratch, maybe you could say that H, it's a symmetric thing. So you could put, I don't know, like the different probabilities between transitions and conversions, or the genetic code. But more importantly is this pi matrix. This pi matrix encodes the equilibrium states of the Markov chain, meaning that at long time, if you start from any given sequence and you let your evolutionary model run for a long time, you will converge to a distribution over states that follows this P1 to PQ. Okay, so for nucleotides, which would be like this, and for amino acids, you would have some distribution over amino acids at infinity. So this is the long-term state of the chain. And so essentially, this Q matrix encodes, in a way, the function. because it gives us the long-term state of the sequence. So obviously, if what's the state of the art is that you take one Q matrix per sequence position. So you consider each position as evolving independently from the others. So you have one matrix per position. And you reconstruct your likelihood for each position independently. And now, instead of having sums over full sequences, you just have sums over the number of states at one position, which say would be 20 for amino acids, right? And then this becomes doable. So essentially you can write everything as products between matrices and vectors. And so this is done very quickly. So essentially, once you have this thing that you have one Markov chain per position, the only question you ask is how do we find once you decide on the Q, and once you have the tree, you're essentially done. So the state of the art is to say okay, you have many methods here. What people use the most, and maybe people who know more in the audience can correct me, but is to use the same matrix Q for all sequence positions. And typically, these are matrices that are pre-learned on some curated data sets. So for proteins, you have the JTT matrix, the LG matrix, and others. And then you assume that the states can change across positions, right? So typically, you would do, for instance, 4 mutation rates, which are gamma distributed, and you you infer the parameter of this gamma distribution. So you essentially allow for different positions to evolve at different rates. An important thing here is that you completely ignore functional constraints that come from interaction between positions, because by design, you consider everything independently. So every position on the long term, if you let this evolutionary model evolve, every position on the long term will converge to the same distribution. Okay, it might take more time for some than others, but they will all converge to the same amino acid distribution. Now the question is, can we somehow improve on this and use some generative sequence model to build a propagator that is more aware on the functional constraints? The problem is that if you use something like, say, a POT model, or maybe some deep model like transformers, is that the probability of a sequence is not factorized. Obviously it's not independent. So you're back to the same problem that you will have to sum over all possible sequences at each internal nodes, and you don't know how to do this. So you somehow need to find a way to take this epistasis into account, but without, with still having this hypothesis of having one matrix per sequence position and doing one position after the other. So this is where the autoregressive model comes in. So here the idea of these autoregressive models is that you write the probability of any sequence A1 to AL. So these are amino acids and the sequence is of length L. You write this as a product over conditional probabilities. Okay, so this is, the first equation here is just the definition of conditional probability. It's something that's it's always true. Okay. The interesting thing is, can you somehow find a functional form P of AI given the amino acids that are before I, and learn these conditional probabilities? And if you manage to learn these conditional probabilities, then you can, sample the first site from something. The first site depends on nothing else. Okay. Then you can sample the second site given the first site, and so on and so on, and you can generate sequences. And so here, what we are using is the results of this paper here, where they found that you can use a relatively simple form for these conditional probabilities, where each site has one coupling to each previous positions and no coupling to the positions after, right? And so you learn these J parameters and these H parameters from an alignment. So you start from a protein family, you learn your parameters here, and you end up with your generative autoregressive models. So note that in general, so the technique that I will describe below could work for any autoregressive model. And we choose to use this particular architecture or this particular parametrization because it's extremely easy to infer from alignments. So it's something that's done in a few minutes, even for very large alignments. And the parameters are somewhat interpretable. So these couplings typically correspond to contacts. So we like it for this reason, but we could use something else in principle. So the idea now that you have this autoregressive model is that given the context, so if I imagine that I know the state of the first I-1 positions in the sequence, then the equilibrium frequency, the amino acid distribution at position I depends only on this context that is before, right? So I know I have all of the information I need to compute the equilibrium frequencies for position AI. And so I can essentially use this in this Markov chain framework that I've described before. So suppose that for all of the tree and for all of the internal nodes, I managed somehow to reconstruct all of the sequences, but only at the I-1 positions. Then for all of the internal nodes, I also have the equilibrium frequencies that site AI, that site I should take, right? And so what I can do is I can derive or construct a custom evolution or transition rate matrix Q that is specific to the position I and specific to the internal node that I'm dealing with, okay? Because each internal node will have a different context. So I'm essentially constructing matrices that ensure that each internal node, so for position I have the information at each internal node of all of the previous amino acids that I've reconstructed. And this has two important advantages. The first is that you still reconstruct one position at a time. So I'm going to do position 1, which is independent of all of the others. And so I use the algorithm that I've described before. And at this point, I have reconstructed the state on the whole tree for position 1. And so I go on to position 2, and so on and so on. And so this way, I'm always dealing with just one site at a time, and so with only an alphabet of 20 amino acids. And the second advantage is obviously that this is aware of the functional constraints, because in the end, the frequencies that you compute come from this generative model. And the cost, there is one hidden cost in a way, is that you need to infer this model. So you need to have the computation time. It's not very long for this one, but also you need to have good enough data to infer the parameters of this autoregressive model. And one thing that I state, because it will be useful for the rest, is that, so I mentioned that it's 20 amino acids. This type of model usually represents the gap state of the alignment as an extra amino acid. So this is kind of the convention in the field. So actually the alphabet that I'm using here is of size 21 because you have the 20 natural amino acids and the gap symbol. So these matrices are 21 by 21. And this, so as we will see, it can be seen as an advantage. I think it's an advantage, but one can debate about this. So how do you test whether this framework is good? Well, the problem in all of phylogeny is that you don't really have data, you don't observe a lot of ancient proteins, or at least not extremely old. So the easiest thing that you can do is simulate evolution using some model. So using a synthetic tree and a model that you've inferred, you simulate an alignment, and then using the tree, your synthetic tree, you try to do ancestral sequence reconstruction. And
Pierre,
so here, this is what we do. So we use the sequence. Yes.
Could I interrupt you for a sec? I was hoping. so it wasn't obvious to me that so we're working in a phylogenetic, maybe you can go back to the previous slide. We're working in a phylogenetic context here. And so just walk us through what are the sequence of events. So we're computing a likelihood, right?
Yes.
So I just want to the form like are we moving up and down the tree, because it seems like we still have to do some sort of Felsenstein sort of thing, or
yes.
Are we,
so yes. No, you're right. So essentially, thing where I compute for each node, for each internal node, I compute in one pass over the tree, I compute the likelihood of the data that is below the node, and in another pass across the tree, I compute the likelihood of the data that is above the node, right? And I do this position per position, so I can do this in reasonable time. And once I have this, I can just, I just have the for a given position and for a given node, I have the likelihood of each amino acid at this node and at this position. And then I can do, so either max likelihood, either I sample from the likelihood, which would be kind of Bayesian. I don't know if this...
Yeah, let's see. Okay, so when you're calculating, so I understand all that for the independent sites thing.
Yes.
Like independent sites.
Okay, so, okay, yes. But this, yes. No, so, in the end, what I do is always, so the algorithm I use is exactly the one for the independent sites, right? It's just that the way I proceed here, so normally if you do independent sites, you could do this, this Felsenstein, you could do L times in parallel for each position, on different computers, and this would work.
Yeah.
Here, what I do is I do it for the first column of the alignments, essentially. So I can elaborate, but let's say for the first column of the alignments, and then I infer what the ancestral amino acid should be for the first column at every internal node. And at this point, I have an evolutionary model for each branch of the tree, but for my second site.
Great. This is exactly. Yeah. So this, and then you proceed your way along.
Exactly. So it's one position at a time. So you use the same. It's just not parallelized in a way, because each position depends on what you've reconstructed before.
And do you propagate uncertainty through, or you just choose one?
No. So we just choose one. No, we haven't found a way to propagate uncertainty. So I think yeah, okay. No, I don't know if I don't think it's possible to propagate uncertainty. Maybe I'm wrong.
I think you're probably right. It would lead to some exponential, right? Because you need to keep track of the full sequence.
Exactly. So intuitively, I think you go back to the same problem if you try to do this. So no, we fix the sites, we fix the context, and then we yeah.
Okay, that really helps. Thanks. Yeah, I just wanted to have it in my head how this all goes. I appreciate it. Thanks.
No worries. Thanks for the question. Okay, so yeah, as I said, to evaluate this, we essentially simulate evolution on a given tree that we synthetic tree, and we reconstruct. And so we reconstruct either with the method I told you, and importantly, the simulation method is the one that is based on the autoregressive method. Or we reconstruct, so here using IQ3, so this doesn't tell you a lot, so we use different methods in IQ3. We essentially let IQ3 decide on the best evolutionary model, so it has a model finder mode, and so we use this. And most of the time, I think it most of the time finds the LG matrix and some gamma rates or some free rates, depending on the specific family we use and on the specific tree. So essentially, we consider this as a representative state-of-the-art method. And since you simulate evolution, then you can just compare your reconstructed sequences to the real ones that you've simulated, so you know them. And the kind of plots that we do are the following. So let me explain. Here on the x-axis, what I have is the node depth. So it's how far away the internal node is from the leaves, okay? So the root of the tree is the deepest node. So the root is not actually here because it's not very well defined as a node in a way. But essentially, the nodes that are closer to the roots are more on the right here, further away from the leaves. And on the y, we have the Hamming distance normalized between the reconstructed sequence and the real ancestral sequence. And this is averaged over many trees. So we do this for many synthetic trees, I think 100 trees of each 100 leaves. So these curves here are average, and you have one sigma here is the error bar. So what I'm showing you here is the ML reconstruction. So we just take the sequence that is the most likely according to each model, which is the thing I think most commonly used in literature. And what you see is that IQ3 typically lands farther away from the truth than the autoregressive method. So this difference gets higher and higher as you go deeper into the tree. So the black curve here is the improvement, is the blue curve minus the orange curve. So the fact that this is positive tells that essentially what we do is better, right? Which is the goal of every method. So this is not completely surprising in a way, because the evolution method that we use is precisely the one with which we reconstruct, okay? But I will come back to this. One interesting thing is that if you look at nodes that are extremely close to the leaves, here IQ3 is still performing much worse than the autoregressive. And this is not very intuitive, because you would think that if you just have one mutation, even if you have an independent model, you will reconstruct it quite well. And actually what's happening here is that in our evolver method, based on a generative model, we have gaps in the alignments, okay? And these gaps are very well represented in our autoregressive method. So they're just one amino acid among they're the 21st amino acid. So our method is able to reconstruct gaps in the ancestral sequences, whereas IQ3 and I think most other packages that exist do not actually model gaps, or at least not with the basic settings. Maybe you have some more complicated models, but in the basic settings, what they assume is that gaps are missing information in a way. And so every time you have a gap, some amino acid gets reconstructed. And this is completely, so in this case, this is completely wrong, because the ancestral sequence sometimes has gaps. And so this is the reason of this delta here, is that we're able to model gaps, I think, in a way that is makes a bit more sense than the current state of the art. So if you say that, okay, this is not initially the effect that we're interested in, the initial intention was to model epistasis better, not gaps, then you can also compute the Hamming distance ignoring gaps. So you ignore every position that is gapped, and you see that here you still have an improvement, which is very small, close to the leaves, and that gets higher when you go deeper and deeper in the tree. So this is something systematic, that epistasis starts to matter when you go further away, so to higher evolutionary times. For small times, it doesn't really matter, but for large times it does. You can also do Bayesian reconstruction. So not in the sense that, just in the sense that you sample from the likelihood. So you use the likelihood as a prior probability. And here you get the solid curves that are here. So the dashed lines are the ones from before, and the solid curves are the Bayesian reconstruction. And here you see that so it gets worse, typically, more further away from the truth, but you still have an improvement. So as I said, in the case before, the evolver that we used was exactly the same as the one we used for reconstruction. So what we can do is we take another generative model to simulate the evolution, and we see if this still works or not. And here we are using a POTS model, so which is, say, a bit more precise and also a bit more well-known than these autoregressive models. And we do exactly the same game, which is to simulate and reconstruct. And here we also get a systematic improvement, but typically much smaller, okay? So we still get, so this is with gaps, this is ignoring gaps. And here you have a systematic improvement of maybe 2-3% Hamming distance, but which is much smaller than the previous one. So what this means is that it's not only the fact that you're using a generative model, it's how exactly the evolution is simulated seems to matter for these reconstructions. And so obviously we don't know what, it's hard to tell what nature actually does. Is it more like autoregressive or POTS? I don't know. But yeah, so there is this caveat here that performance depends on the type of evolver that you use. But still, the fact that we integrate functional constraints gives a systematic improvement. So another thing we did, which I think is quite interesting, is to use or try to use experimental data for this. So here we used an experiment of directed evolution, where so it's this paper here, where they took beta-lactamase protein, so PSE1, and they introduced, so typical directed evolution, they introduced mutations, then they did selection in vivo for antibiotic resistance, and then they take they take a sample from the population here, they reintroduce mutations, and they do rounds of this, right? And so you have many generations. Some of them they sequence, others not. And so here we are interested mostly in generation 20. So I think they sequence generation 10 and 20 and maybe another one. I don't remember. Essentially, what's interesting is that at generation 20, you start having a good variability in the alignments. And what we're saying here is that in a way, these sequences at generation 20, we know their most recent common ancestor because we know what sequence they started from in the experiments, okay? So obviously the problem is we don't observe the tree in these experiments. So you have to assume something. Here we did a very rough simplification, which is to assume that the tree is essentially star-like. So you have very few branching events in the tree and they all converge directly to the MRCA. So this seems very rough. At the same time, the population size is quite large. So you don't expect too many coalescence events close to the leaves. So we hope that this is good enough of an approximation in a way. And so then what you do is you take this alignment and you try to reconstruct, and you can reconstruct, so either with IQ3, so here it was the JTT matrix plus G4, either with the autoregressive model, and another thing we tried is to just take the consensus of the alignments here and say that the MRCA is just the consensus of the alignments. And so here are logos of the reconstruction. So there are a lot of sequences at generation 20, maybe 10,000. So it's a bit high for our algorithm. So what we do is we subsample these alignments. So we do sub-alignments of 100 sequences, I think. And we do different reconstructions for different sub-alignments. And then we look at how frequently each character is reconstructed. And there are essentially 6 positions where the consensus reconstruction fails. So here, the consensus should always be different from the wild type for all of these 6 positions. And now if you look at IQ3, it gets, I think, 2 positions better. So it gets this I in the first position, IQ3 fixes, and I think another one, which, this Q here, it finds now that this should be a Q instead of an L. And now if you do the same with the autoregressive model, you again gain essentially 2 positions better. So now it also finds this L here, and it also finds this A in the 4th or 5th position across these. So, this is obviously, this is one data set. You have this very rough approximation. So, I don't want to say that this proves that the autoregressive will always be better, but I think it's an interesting way to use experimental data for phylogeny. And it's quite encouraging that using the functional information, you do manage to actually improve in an experimental setting this reconstruction. So another thing that I want to talk to you about is the biases of the maximum likelihood reconstruction. So for now, all of what I've shown, or most of what I've shown, is to just take the sequence at each internal node that maximizes the likelihood. So you take each amino acid that maximizes the likelihood. So this is the method that's most commonly used in literature. And experimentally, so there are many papers where people have tested this. The sequences that you reconstruct this way typically are functional and often highly thermostable. So more thermostable than the present-day proteins. And obviously, there is one problem here, which is the one of biases. Essentially, is the most likely sequence representative of the ancestor, right? So you know that your most likely sequence will still be, at Hamming distance, maybe 20, 30% from the real ancestor. So depending how deep in the tree you are, but it will always be significantly away. And so what you could say is it's maybe not the best idea to take the best sequence, but one should rather use a representative set, which would be less biased. And so this would be, for instance, by sampling from the likelihood, so sampling from the posterior probability. The problem is that instead of having one sequence at each internal node, you get a set of sequences at each internal node. And so obviously, if you want to experimentally test them, it's not as easy. So this is not very often used in practice. And it turns out that, so sometimes you get functional sequences, but in some cases, you get sequences that are not functional at all when you do this. So it kind of depends. And so the question that we can try to answer here is that, is this Bayesian set somehow more representative of the real ancestor? So why do I think that we can address this question? Here, we do simulate evolution with a generative model, which means that we can actually compute the probability of each sequence according to our generative model. So for each reconstructed ancestor, I can tell whether my model likes it or not in a purely generative setting. And so what we will do in a way is that we will consider this probability, or the log of the probability, as some kind of proxy for function. So sequences that are very improbable in the model should not be functional. And we want sequences that have a typical probability in the model. So the kind of thing that we can do is measure the log probability of the reconstructed sequences as a function of how deep we are into the tree. And so this is done here for the three methods. So for IQ3, so PMB matrix plus independent sites plus G4, autoregressive model, and the real sequences. So this is using simulated data, so I know what the real ancestors are. And I can, again, compute the log probability of all of these reconstructed sequences, depending on how deep they are in the tree. And the dashed line here is the average log probability that you get if you sample a large amount of sequences from the model, or equivalently, if you take the alignment at the leaves of the natural sequences of this family, the average log probability is the one that you have here on the dashed line. So it's representative of a typical set of the model, if you want. And so here, what we see is that if we do the maximum likelihood reconstruction, both autoregressive and more impressively IQ3 tends to go, so the reconstructed sequences by both of these methods tend to increase in log probability as we go deeper into the tree, so as we go further away from the leaves. So this is not completely surprising, because when you ask for the most likely sequence, well, the most likely sequence somehow integrates maybe information of this model through the propagation, so through the evolutionary model or through the leaves. And so you expect the sequences that you reconstruct to be more probable according to this one. The thing is, this completely deviates from what the real ancestors are. So the real ancestors stay in this, typical range of probability of the model, which is expected because they're simulated with this model. Another way to see this is you can here see the distribution of log probabilities for a sample of the model. And here I put the reconstruction of the deepest nodes as bars, okay? And we do many repetitions of this. So this is for 100 different trees, the log likelihood according to the model of the deepest node. And you see that the real deepest node is typically in this range. And if you take the reconstructions, they tend to be biased to more likely sequences. So what I mean by this is that ML reconstruction is biased, and you can measure it using these generative models. And what it's biased in the sense that it generates sequences that are too good according to the model, in a way. Okay, so they're actually unlikely in the sense that they have a very high probability, but you're unlikely to see them in a sample because they're actually too good. And what you can do is you do Bayesian reconstruction. And here we observe something very interesting that we don't quite know how to explain, which is that if you do Bayesian with the autoregressive model, which is the one that was used for simulation, then you completely remove this bias. So it's the dashed line here. You stay in the range of typical log probabilities of the autoregressive model. Whereas if you do the same using the posterior probabilities that the site-independent model, so IQ3 gives you, you still have this very strong bias. So you still generate sequences that, in a way, are too good. And so we kind of understand why it's doing this, but not quite. And I will detail a bit on the next slides. But essentially, the conclusion here is that maximum likelihood is biased. And if you want to really have a good idea of what an ancestral sequence was like, it's better. So obviously, it's easy to say as when you're doing simulations, but it's better to simulate an entire set of sequences, and this will be more representative. So this is what I'm saying here. So you have a strong bias of log probability for ML reconstructions. And actually what we see is that this bias can be explained, or at least there is a correlated observation, which is that the deeper you go into the tree, so the further away from the leaves, the more the reconstruction resembles to the consensus of the alignments, okay? So here the solid lines are ML reconstructions, the dashed lines are Bayesian reconstructions, and so you have the same color code. The green is the real sequences, orange are the one with the autoregressive model, and blue are the site-independent model. And so you see that the deeper you go into the tree, the closer you get to the consensus. But for the site-independent model, or for the maximum likelihood autoregressive, you get much closer to the consensus than what you actually should be doing, right? So you don't find something that is very representative. You just converge to the center of mass of your leaves, in a way. And so this is a clear bias. And I think it probably explains, even though I don't have very good data to back this up, but my intuition would say that this explains the high thermostability that people find in ancient proteins, is that actually, you get closer to the consensus, you get to sequences that have all of the best amino acids at the best positions, and this ends up being very thermostable. But maybe it's not actually like this. Maybe the real ancestor was actually more typical. Okay, and so I jumped this because it's actually a bit technical. So I jumped the detail, but I just mentioned it if people are interested. There is one caveat with this autoregressive dynamics, which is actually quite important, which is that they're not Markovian, which is very unexpected. But so the detail is a bit more technical, so I kind of leave this for questions if people are interested. And I jump to the conclusions. So essentially, I've shown you an evolution model based on a generative model. So we're taking advantage of the autoregressive architecture to have something that's almost factorized. It converges at long times to a generative distribution, which is something that site-independent models cannot do. It does have some caveats. And when we do ancestral sequence reconstructions with it, we find that it has a better handling of gaps, or at least that's an opinion, I think, but I think it handles gaps better than what current models do. It does show a systematic improvement over state-of-the-art models in simulations, and seems to also improve when we use this directed evolution data, which is encouraging. And an interesting thing is that we can also use this framework to study the biases between ML reconstruction and Bayesian reconstruction. And with this, thank you very much for listening. And if you have any questions, I'm happy to answer.
Excellent talk, Pierre. Thank you. That was very clear. Well, so I invite anyone to ask any questions. I'll start with maybe sort of an obvious one, which is like you currently have an autoregressive model that goes from one side to the other.
Yes.
And then you saw with your POTS model simulations that maybe, well, I don't know, I guess I'm hypothesizing here. Maybe the fact that it's autoregressive is one of the things that's making it difficult for you to match the POTS models in simulation. And I just wonder, like, I mean, I don't know, it's like, obviously it's not possible to like do an autoregressive model conditioning in both ways, but like, could you do something stupid and heuristic where you sort of do the autoregressive model in one way, do it in the other way, and then do something where you're sort of like taking maybe like the most likely, like compare the predictions between the two different directions and then see if you could sort of make a best guess as to what the site is?
So just to clarify, when you say in one direction or the other direction, you're referring to the fact that to this
in the
towards
upper right, you have like AI conditioned on A less than I, but you could also do it in the other direction. You could have AI.
I see. Okay. No, so this is a point. Okay, this is an interesting question because this is something that I put under the rug for to have an easier presentation. So the order of the ordering of sites here doesn't actually have to be 1 to L, as you say. It also doesn't have to be L to 1. I can actually choose any permutation of the sequence, and you start from any site, then choose any other as the second one, and so on. So when they inferred, so in this paper here, they had this question, obviously, which is, we want to choose an ordering that makes sense. And what they found is that the best performance in terms of generative capabilities is to first use the most conserved sites, then the second most conserved, and so on and so on. And the last one you deal with is the more variable. So there is no theoretical explanation for this. I think it kind of makes sense, but it's mostly intuition. And so here, what we did is we just went along with this. So we said, okay, they found that the best ordering is this one, and we're just going to use this one. What you say is actually very interesting, is how much the dynamic depends on in which, how you order your autoregressive model, right? So this we played a bit with, and it does depend. So for instance, if you decide to start with the end and go to the first, or the first to the end, or do this conservation order that I mentioned, you do get dynamics that are a bit different. The problem is then you have L factorial possible arrangements of this autoregressive thing, and we don't really know what to do. So, I mean, interesting point, but I don't really know how to answer this, because there is an infinite amount of things that we can try, and we don't have a very good theoretical thing to guide us.
I guess this is, again, silly idea, but I mean, you could take several different orderings and then you could look at which one of those is most confident in its prediction, and then take that.
Yeah.
I don't know.
No, yeah, this could definitely work. Yeah. So the main issue here is that anytime you take a different order, and you also need to re-infer these parameters of the model, right?
Right. Yeah. Okay.
So it's not. I mean, you can definitely do this, but it quickly becomes computationally intensive, right?
Yeah. Okay. Well, maybe I'll ask a second obvious question, which is, I mean, there's a lot of autoregressive generative models for proteins out there that aren't. I understand what you're doing is different. Like, you're training a model that's specific for a given sequence alignment, but you like I sort of wonder if you could take the same framework and then use the protein representations, or do some fine tuning or something like that in order to.
So the fine tuning I haven't thought about. Probably you could definitely take something like ESM or any large language model on proteins, for instance, that has autoregressive capabilities, and use, I believe, exactly the same algorithm. The only thing that you need is to be able to compute this with that, and I think that you can with typical large language models.
Well, let's see. So they come in two flavors. Like, there's things like Progen, which are autoregressive, and I don't think ESM. I think ESM is.
Yes. So for the learning, definitely not. But you're sure for the generation that they're not autoregressive?
I mean,
okay. Maybe I may be mistaken.
Well, it's totally fine. I don't know. I mean, that was just my impression, but that's. But anyway, okay. So you've thought about this, and.
Yeah, I mean, we haven't tried yet, but at least in principle, it's something that you can do.
I'm loving the fact that you can do indels in your model, and I guess it sort of makes me want this also on a nucleotide level, and I wonder if you could do something where you're combining. I mean, so the autoregressive part is going to make a lot more sense on the protein level, but I guess I wonder if you could combine it with a codon model, so that you would get basically like a model that works in nucleotide land, but leverages all the autoregressive stuff that's happening in protein land.
So we did not do this at the level of nucleotides, but we did include some terms that come from the genetic code. So what I said is in the beginning is that these matrices, you have one part that is the equilibrium distribution, and this is the part that where we put the autoregressive probabilities. And you have one part that is a symmetric matrix, where you can put any symmetric matrix with positive terms, and you will have a Markov chain. And here, what you can do is you can simply, in this H matrix, you can put values that are proportional to the number of possibilities to go from one amino acid to any other, right? So something that takes into account the genetic code. And so it's not at the level of nucleotides in the sense that you still don't reconstruct nucleotide sequences, but at least you take into account the nucleotide basis. And this turned out to be much, much better for the experimental data. So here, clearly, there was a big difference between using this and not using this, which makes sense, right? Because you start from one specific sequence here, and in the end, this is relatively few generations. So you have some stuff that is inaccessible because of the genetic code, and so this was so yes, you can do this.
Yeah, that's cool. That is different than the sort of thing I was thinking about, which is more along the lines of, let's say we have trained our autoregressive protein model. You could then come, if you wanted nucleotide sequences, you could come back, and now that you can calculate protein likelihoods, you could come back with a nucleotide model, like a codon model, and then assess the likelihood of the different like you could use an HKY for the nucleotides, but then sort of like wrap that into a codon model and then figure out what the ancestral nucleotide sequence is.
Yes. So you would essentially infer codon by codon. With, yes, as you say, some simple nucleotide model. Yes, one could do this. I think, yeah, this would be possible. I haven't thought about it, but yeah, mostly you just translate. Yeah, I think it would be possible.
Anyway, and I guess I was going to ask about the side that you skipped, but I think what's going on is you just have like mutation effects are propagating farther than just a single nucleotide, right? I mean, you have some change, and then that propagates to the next site, and then because of that, it propagates to the next site, and that's what's not Markovian.
Yes, it's mostly this. So actually, one way to explain it is that you can say you mutate this site, okay? Then maybe your model says that if you mutate this site, the next amino acid also needs to change because it's very bad. It's a very, very epistatically constrained pair. But in finite time, this one doesn't always change. And you're considering this completely independently. And so this means that sometimes you have like very bad mutations that your model does not account for. So it's more or less what you say. So this is something we figured out actually at the end of this project. It turns out that quantitatively, it's not a very strong effect. So we do see that it's not Markovian in the sense that you start from a sequence and you sample a bit, and so you sample for a long time, or you sample for a short time, you stop, you sample for a short time, you stop, you do these kind of things. And this should always be equivalent if you have a Markovian process. And here it's not completely equivalent. Your distributions shift a little bit, but it's kind of an order 2 effect. Yeah. And I think it's partly because of this that we don't match the POTS model so well. I think also because I mean this would be my intuition.
Interesting. Yeah. Well, we're at time. I really appreciate your talk, and yeah, good luck continuing. It's really exciting.
Thank you very much. Yeah, thank you for inviting me.
Yeah, have a good one.